Okay, remember, by the way, Pear Park is tomorrow. So if you're still thinking about wanting to go to there, please, by all means, you are invited, of course. Uh, the directions and all the information are on Blackboard to take a look. And if you have any questions, just give me a holler about that. All right, so what we're going to do now is go back to that overall, overall origin of life on Earth, and talk now about the progression of life on Earth. So once life evolved, once that whole biogenesis happened, and remember we talked about that in class, and I mentioned to you that I'm giving you very broad sort of ideas, or very broad sort of concepts that we think must have happened in order to lead on to this idea of life. And again, whether it happened that way exactly or not, at this particular point, we certainly don't know, and may never know. But we know, as far as, if life exists the way it does today, then we're thinking that these are the things, given the conditions we knew what it was like on Earth, these are the things that must have happened that led to this complex chemical reaction, this thing called life, right? First, you got to have the organic molecule. Carbon works well for that because there's lots of places you can bond it together. Then you're going to make bigger molecules. And the important thing about the macromolecules, again, folks, was especially when you think about proteins, macromolecules, long proteins, means with many different side groups, you can get lots of different shapes. And proteins are important for living organisms because of the fact that you can do lots of things with them. There can be structural, functional, enzymes, transport proteins, all those things you learned about in Bio 1. And then the next step was the idea of packaging those special molecules that can do these special sort of metabolic tasks, put them inside of a protocell, a phospholipid bilayer, and that concentrates the ingredients in there, or concentrates the materials. And when you do that, of course, then there's a better chance for them to come into contact with one another. Remember, we talked about that in class. And maybe in Bio 1 you did too. Enzymes and substrates get together. I mean, they fit because they have the shapes, but they get close enough to even fit because of randomness, randomly moving around until they bump into one another, essentially. But if you package them in a smaller volume, then I think I gave you the analogy of two people in a gym running into each other versus two people in an elevator. You got a much better chance of these two things coming into contact with a smaller volume, okay? And then after that, the last thing was some way of remembering how to make these macromolecules, remembering how to make those proteins, and remembering which amino acids in one order, and they come in one order. And that information is all going to be retained in some self-replicating information storing molecule. And you recall the first molecule that fit that bill, we think, was probably RNA, not DNA. DNA, again, remember, is sort of the permanent archive of the information. If you recall from Bio 1, DNA is never used in any way other than for information. Now, you make copies of DNA when cells make copies, but other than that, when the DNA, during transcription, you unwind the DNA, and then the RNA polymerase reads the DNA to make the messenger RNA, but then the DNA goes back together. So the DNA stays DNA, and it doesn't leave the nucleus out to a ribosome, any of that sort of stuff. It's the permanent code. So it's the archive of the information of how to make these special molecules, okay? All right, so let's take a look at this. I love this little clock. I was talking to you guys the other day about trying to figure out all of this progression of life on Earth as humans, as us coming in at the very last. Right here we are at the very, that last little brown part up here at the top, and that's the human being. So after all of this has been going on, us human beings enter onto the stage at the very, very end of all of this, and we're trying to figure out what's been going on for 4 billion years. That's quite a task. So this clock here is going to break it down into sort of the major things that went on, and then we're going to look at each one of those and find out specifically what was happening. So back about 4.5 billion years ago, like we talked about, the origin of the solar system and the Earth came about. Planets and the sun and all the planets of the solar system and the sun, hold on, I'm wrapped up in my mouse cord there for a second, all evolved or originated at about the same time. Not a lot was going on the Earth for about 1 billion years, because remember, the Earth was this nasty place. It had a lot of volcanic activity. There was volcanic eruptions, hot rock, magma, all kinds of poisonous gases, hydrogen sulfide. all those gases that were good for making complex or simple organic molecules, right, that we talked about last time, but not particularly good for living organisms, okay? So it's about 1 billion years from the time the Earth began until we started to see the prokaryotes. And they would have been the first ones, probably to no surprise, right? They're the relatively, again, I don't want to insult prokaryotes out there, but they're relatively simple, let's say ancestral type critters. There's not a lot to them. They're alive, which is special enough in its own right, but, you know, they're fairly basic when it comes to living. Now, what happens next, or one of the next major things that happens, is some of these prokaryotes were photosynthetic. And if you remember in the early Earth, I said, you know, we didn't have oxygen in the early Earth. That's why we were able to actually have that reducing atmosphere. So photosynthesis was a way for organisms to make food. It still is a way for organisms to make food, but back 3.5 billion years ago, that was really important, wasn't it? It was really important that critters come along that can photosynthesize. Now think about that for a second. Why is that such an important key event to occur to life on Earth, that an organism could photosynthesize? Why, other than what you see there with atmospheric oxygen, before that, why would photosynthesis have been an important metabolic activity to arise in organisms? Think about the first critters on the planet. What did they eat? You know, the first little bacteria swimming around, what did they eat? Unless you were like one of those special ones that we talked about, where you can eat the iron off of, let's say, the Titanic, most of the organisms back then would not have had anything to eat. So we think that, right, photosynthesis would have had evolved pretty quick, because photosynthesis doesn't require you be in a place where there's food. All it requires is you be in a place where there's water, carbon dioxide, which there was plenty of those two things in the Earth, and sunlight, which there was plenty of sunlight back then. So some of the first critters on the planet would have been those photosynthetic prokaryotes, and then there would have been other prokaryotes that arose that would have fed upon those other organisms, and then there would have been other prokaryotes that can make a living in those non-conventional ways, like through chemosynthesis, you know, down around those geothermal vents that I was talking to you all about. So there wasn't just photosynthetic prokaryotes, but photosynthesis, the actual chemical process of photosynthesis, is the splitting of water. So the gas that's given off when you split water is oxygen gas. And you learned about that, I know, in Bio 1. Most of the organisms that we're familiar with on the planet today are oxygen-breathing organisms. They're aerobic organisms. They need oxygen for reasons that you learned about in Bio 1 in respiration. You remember, if it's aerobic respiration, how many ATPs do you get from aerobic respiration versus anaerobic? Anybody remember? How many ATPs do you get if you take one glucose and it goes through the entire process of respiration aerobically? A little more than 36 to 38, yeah, somewhere around 36 to 38, Jennifer. That's right. How many do you get if it's anaerobic? If you only do glycolysis? That's it, just 2. So right away, you can see that there would have been a distinct advantage to being an aerobic organism, right? Because from the same glucose molecule, now those would have been the photosynthesizers that made those. That's what happens in photosynthesis, remember? But from that same glucose molecule, you could get 14, 16, 18, 19 times more energy, 36 to 38 versus 2 ATPs from the same glucose. Now, we've gone through a whole month of talking about evolution. Do you think that being able to do that, to get 17, 18, 19 times more energy out of the same glucose, do you think that'd be an advantage? Do you think that would give organisms, you know, reproductive? Yeah, absolutely it would, right? You're more efficient. You're extracting a lot more energy from the same glucose, you know, close to 20 times more energy from the same glucose molecule. So while the prokaryotes, and some of them are aerobic, but in many cases early on, it was the anaerobic ones. When the oxygen began to rise in the atmosphere, then it drove this evolution of organisms that could take advantage of that oxygen. Remember, plants make oxygen just as a byproduct. Actually, it's better to think about plants don't make oxygen. They just take everything off of water. They take the hydrogens and the protons and electrons associated with those hydrogens and leave behind just the oxygen. But once that oxygen began to build up the atmosphere, then, and once aerobic respiration came on the scene, then, boy, things really took off, right? Because now you had organisms that could extract more energy out of the same glucose. And if you could extract more energy, you could become more efficient, you could become bigger, you could become more complex, you can become all the things that we know happened since then. See, prior to this time, we're dealing with prokaryotes. But once this atmospheric oxygen began to rise in the atmosphere, we started to see really this takeoff of these complex critters, these single-celled eukaryotes first, like the protozoans, and then the multicellular eukaryotes. And then, of course, if you jump ahead, now we got the animals and the colonization of land by plants. Then the animals moved out on the land. Again, it's the feeding thing. You can't move out on the land unless there's something there to eat. So if you're an animal and animals cannot photosynthesize, you've got to have something out on land to eat. You got to have plants out there. So they would have been the first ones to actually move out onto the land, would have been the plants. Actually, you'll learn today it wasn't just plants. Plants did it with fungi. They did it together with the fungus. And then we move through the Paleozoic, the Mesozoic is the dinosaurs, and then the recent time is the Cenozoic. Now, as I mentioned, when exactly did life begin on Earth, or, life that we can identify as life, not the biogenesis. So after that, well, we're not really sure because of the fact that these things don't fossilize well. But we have no clear fossils. We have those that may be fossils, those things that are debated, kind of like I showed you that picture from the meteorite from Mars. Is this a geological formation that's formed, or is this actually the casting of some ancient prokaryote or some ancient critter that lived on Mars? But once we have that, those organisms evolving and dying, those are soft, small, soft tissued organisms, soft organisms, not tissue, excuse me, wrong word. They're soft critters, so they don't fossilize well. So there's no clear fossils before about 3.8 billion years. I mean, that's pretty close back to the beginning, but we don't, and life may have begun more than once, started and stopped, but we don't, as far as this time with life, we don't have anything really clear in the fossil record before 3.8 billion years. It'd be cool if we can advance our paleontological techniques to the point where we might be able to distinguish evidences of living organisms that have been fossilized at about that time, reset that time about when it began. What amazes me talking about this stuff is just the time, right? 3.8 billion years. I throw that out there. 3.8 billion years, and about a billion years later, and yet you think about your own life, right? We live 75, 80 years on average, and we're talking in billions of years. You can see why it was so important that Darwin and the early evolutionary biologists to really understand what the geologists were saying about the age of the Earth, right? Because none of these processes that Darwin and any of the other evolutionary biologists came up could have happened that fast. We've got to understand that the Earth has been around a long time. And it's sometimes difficult for beginning students, to kind of visualize this ability from a universal common ancestor, a single-celled prokaryotic critter, into all the diversity we have on Earth today, and all the diversity we've had on Earth, because a lot of it's gone extinct. But again, I'm talking billions of years, folks. So 3.8, 4 billion years, about 4 billion years of that has been going on. All right, so 3.8 billion years ago, no clear fossils. Why? The world was not a very nice place, right? The Neo-Earth was very violent. There was a lot of asteroids. It was a pinball gallery. The entire solar system was a pinball gallery. There were planets and rocks bouncing off each other. Recent evidence even suggests that initially Jupiter formed close in near the sun, about kind of where we are, and then for whatever reason, it migrated out to the further reaches of the solar system. Well, as it was doing that, it was destroying things, this big, massive planet moving through these rocks, and it was just shaking up all kinds of things. There's a suggestion that that's why Mars is smaller than Earth. If you go from Mercury to Mars, you got Mercury, it's real small. Venus is a little bigger. Earth is a little bigger. These are the rock planets. Those are supposed to be round, excuse me. And if we are following what would look like a pattern, we would expect Mars to be a little bigger than that, but it's not. As a matter of fact, Mars is smaller than the Earth. Right past the orbit of Mars is the asteroid belt. And the asteroid belt past Mars is just a collection of leftover parts, they say, from the formation of the universe. Those leftover parts in the asteroid belt, some people believe, could have been what was going to be used to make Mars. So those rocks would have been coming together, forming these planets. But because Jupiter moved out during that time, it scattered all those rocks all over, and Mars never got to be as big as it was going to be. So it was a really interesting place in the early solar system, but it wasn't a very consistent place. So if life was going to evolve during that time, it was going to have to be able to deal with these really horrible kinds of conditions. The heat, the asteroid. No oxygen means there was no ozone layer. No ozone layer means UV light, which UV does penetrate, remember, but too much UV is bad. The early Earth, even before we saw in that picture before, didn't have any water because of the bombardment, because of the asteroids. Water would evaporate. Of course, eventually the Earth cooled off and water began to collect, which there's all kinds of interesting theories about why that happened. The one I like is perhaps comets brought us water. I think, man, it must have been a lot of comets. But somewhere around 3.8, 3.9 billion years ago, these conditions begin to change, right? Life or Earth begins to cool off, liquid water begins to form, and it's at that time that maybe living things begin to show up. And we might not be able to see them again, but we think with the conditions being the way they were, especially with liquid water, we always get very excited about liquid water, right? We find liquid water on planets and another part. We're all very excited at astrobiology because we know life, as we know it, evolved in water, so maybe it evolved in water elsewhere. Okay, so once life began, as you can probably imagine, the two biggest players at the beginning were the biggest player were the prokaryotes, the two domains, and they very early in the Earth's history dominated, the Earth and continue to dominate the Earth. They've actually dominated 3/4 of the Earth's history. The quarter they haven't dominated was the quarter before living things could actually survive on Earth. And very quickly diverged into two domains, the domain archaea and the bacteria. Now that's interesting because we were talking about prokaryotic organisms, and the difference between archaea and bacteria, even though they're small prokaryotic, is quite wide. That's quite a big difference. Big enough that you have to argue that the difference between archaea and bacteria is kind of the same as the difference between those two groups and the other domain, the eukarya. So again, we get caught in this idea of identifying things with the morphology discussion we had in evolution, and we say, well, they're small, they must all be related to one another because they're small little things. And we find out as time has gone on, like when I used to first teach this class, there weren't two domains. There was one kingdom of bacteria called the kingdom Monera. You might even see it in some old books, kingdom Monera. We used to lump all of these together, but now we know that's significantly different. Now, the Archaeans, when we get back and discuss, did we finish the prokaryote chapter? No, we didn't. The Archaeans have an archaic. These are the bacteria, or I'm sorry, the prokaryotes who we think may have been some of the first earliest ones on the planet because they can live in really extreme conditions. There are some bacteria that can live in extreme conditions too, but the high heat, salinity, those kinds of radiation, those kinds of things that would have normally destroyed other living organisms. There are many archaean prokaryotes, and again, there are some bacterial prokaryotes, but really prominent among the archaeans who can survive in these really horrible conditions. Yeah, Thomas, you might have remembered Monera if you had a science class from, I don't know, a few years ago. The name has been changed because, of course, like I said, we've elevated into domains. The more we learn about organisms, I guess the more we learn what we don't know about them. All right, so we got these early prokaryotes, and you know, they've got to find a way on life to survive. They've got to find some way to either obtain food or to make their own food. So some of the earliest prokaryotes, and these might have been the ones that were living down around the geothermal vents, the ones that I was talking to you about last week. Some of the early prokaryotes, what we call chemoautotrophs. A chemoautotroph, you may have seen the word autotroph in Bio 1. An autotroph, troph is to feed, auto is yourself. A chemoautotroph feeds itself and uses chemicals by which to make the food. Okay, so it's making its own food, it's autotrophic, and because if it's a chemoautotroph, it's using chemical processes. They jump down to the bottom of the screen, you'll see another word that maybe is more familiar, the photoautotrophs. Photoautotrophs use light to make their own food, so that would be plants, right? So some of these other critters could have used things like taking carbon dioxide and hydrogen, plenty of that in the atmosphere, and making methane. These are actually organisms that we call methanogens. And methanogens use that very simple chemical process there to release energy, to use energy for their own metabolic needs. You could oxidize sulfur or hydrogen sulfide, another gas that would have been very prominent in the early volcanic Earth. And then I mentioned to you this guy in the picture, which is Halomonas titanicia. On that right? This was a critter that was discovered only on the Titanic, hence the species name Titanicia, if I did spell that right. But that's what that prokaryote is in the picture. And the picture down below, of course, is the Titanic. These organisms can actually oxidize iron, so they're living on the iron plates of the Titanic. So they could eat basically metal, basically an inorganic material that can oxidize it. Now, oxidation, if you remember from Bio 1, is the energy is released during oxidation, energy is needed during reduction. And again, if you have a hard time to remember which one is which, remember reduction is when you're building up a molecule, you're adding electrons to something, so you're building it up. And anytime the laws of thermodynamics, anytime you build something up more complex from something simple, then you need energy to do that. Now, when you break up something that's more complex to simpler things, like if you oxidized hydrogen sulfide and you broke that molecule up, you would release energy. Okay, so however you do it, there had to have been some way to obtain energy to drive those very special chemical reactions that were evolving, as we talked about during biogenesis. So some of the early prokaryotes were chemoautotrophs, and some were photoautotrophs. And photoautotrophs would have been the ones that would have taken advantage of all the sunlight that we had. So the chemoautotrophs, maybe they evolved down around the geothermal vents, maybe around some of the volcanic activity. Maybe there was chemoautotrophs and photoautotrophs evolving at the same time. Nobody knows for sure. But photoautotrophs would have come on the scene, and they would have been able to do photosynthesis. Now, let's go back for just a second and think about what we talked about during our cladistics and phylogeny and systematics, and then we did the little exercise where you had to, you know, drop the cladogram and the different traits that arose. So let's take a look at that and use our technique of parsimony, which we introduced back then, to answer a question. Here is two possible ways that the process of photosynthesis arose, and photosynthetic species arose. There's one on the left, and there's obviously one on the right. So we have an ancestral prokaryote, and then it branches off into different organisms. And the ones that are green are photosynthetic, and the ones that are brown are not. Now, looking at those two possibilities, and remembering what you learned about photosynthesis in Bio 1, and specifically, you learned the complexity of photosynthesis in Bio 1, which one of these two schemes here do you think makes more sense? Do you think there was an ancestral prokaryote and then several different lineages came up with photosynthesis? Or perhaps that ancestral prokaryote was photosynthetic and other species lost the ability to photosynthesize, and that's how they became heterotrophs. Which one do you think is the easiest? Which one is the most parsimonious? Why do you say that, Candy? You're right, but why do you say that? Anybody else? Help her out. Candy's correct. It would have been the scheme on the right. We would have had an ancestral prokaryote that would photosynthesize, and then the heterotrophic ones would have lost the ability to photosynthesize. Yeah, right? So if you lost the ability to photosynthesize, you're not independently creating this complex process like you see on the left. You're just losing a complex process, and that's a lot easier to do than to gain. So yeah, less probable is the fact that evolution or photosynthesis evolved more than once. More probable is photosynthesis would have been one of the first things that evolved in these critters, and then other critters lost the ability later on. And if you think about what I said about the early Earth, it makes sense, right? The first critters on the planet had to have some source of energy, had to have some source of food. And if there wasn't any food around, kind of like I was mentioning about moving out on the land, plants would have been the one that could have bridged the gap because they don't need food. They make their own food. All right, so you got these guys photosynthesizing, right? That's where we are. We have evolution of life, and then we have organisms that are photosynthesizing. Chemosynthesis as well, but photosynthesis would have really taken off because there's a lot of sunlight out there. There's not a lot of those geothermal vents. So there's a lot of sunlight, so photosynthesis would have probably been back then, the predominant way in which organic materials were made, in which food was made. So, and remember in photosynthesis, what's happening is oxygen is being produced. So we go back to these organisms we looked at under the lab the other day, Anabina and Oscillatoria. You remember those two? Your lab manual calls those cyanobacteria. Now, cyanobacteria literally means blue-green bacteria. Sometimes they're referred to as blue-green algae. Let me say something about this word algae for a second before we go on. There are many different things that are called algae. For example, Anabina and other members of the prokaryote group there are known as blue-green algae. They come a blue-green color to them. There's also a plant algae. There's also what we call an algae that's actually a plant. It's not a prokaryote at all. So when we're using the word algae, we have to be careful as to what context we're using it in. Is it a prokaryotic blue-green algae, or is it a is it a green algae or a brown algae, which in some cases is a plant? And then there are even some things we call algae that aren't plants at all, or protozoans. For example, seaweed. What we call seaweed is not a plant; it's actually a protozoan, and it's actually considered an algae. So. Got to be careful with that term. But these blue-green algae were producing oxygen, and they made massive amounts of oxygen, and they grew in these huge mats of cyanobacteria. So like the Anabina and Oscillatoria that you saw, they would grow these huge, gigantic areas they would cover. Stromatolites, in the lower right are some stromatolites. I think these are from, I don't have my book. I think these are from Shark Bay, Alaska. But what a stromatolite is the mineral materials that were accumulated by these mats of photosynthetic critters. And then through time, what has happened is these are right on the coast. The ocean has moved in and has eroded through those stromatolites. And that's why you see, so imagine all those lumps that you see in the picture there were one solid sheet, and then the ocean kind of eroded all those lumps that you see there. But what is significant about the stromatolites is how thick and how big they were, but because of the fact that we had lots of photosynthetic things. So once photosynthesis kind of came on the scene, and with this sunlight that we had, oh my God, everything went crazy with the photosynthesizers. There's tons of them. It's a great place for photosynthesizers to live. And so the oxygen levels begin to rise. okay? And the oxygen is being given off by these photosynthesizers as a byproduct, right? They're not making, the organisms can't make that with the idea that, oh, someday perhaps a critter will use oxygen. First, the oxygen levels begin to rise in the water. A lot of these algae are going to survive in liquid environments. First, it's going to rise in the water. First, it's going to combine with the iron in the water, so it's going to make rust, okay, basically. But after a while, the oxygen is going to saturate the ocean, and after that's done, it's going to begin to diffuse into the atmosphere. Right? It's going to begin to move from the atmosphere, from the water up into the atmosphere. Now, for some anaerobic prokaryotes, that was their death knell. That was the end. Many anaerobic prokaryotes cannot live in the presence of oxygen. There's still some around today that are that way. There are still some what we call obligate anaerobes. Obligate means you have to do anaerobic respiration because they're also facultative anaerobes. Facultative anaerobes can do both, anaerobic or aerobic. But many obligate prokaryotes, obligate anaerobic prokaryotes, die in the presence of oxygen. Remember, oxygen actually is a bad molecule. It tears things apart. It oxidizes things. So the organisms are particularly susceptible to that. So being around atmospheric oxygen would have killed them. So a lot of these early prokaryotes would have died off. Others found a way to survive, which I'll talk about here in a couple slides, something known endosymbiosis. Others found these really neat mechanisms by which they could survive these rising oxygen levels. About 2.2 billion years ago, the amount of oxygen on Earth rose dramatically, and that's when we start to see eukaryotic algae. All right, so there's blue-green algae, now we got eukaryotic algae. In this case, the eukaryotic algae could have been, it could have still been prokaryotic, but it could have also been, I'm sorry, not prokaryotic, it could have been plants or could have been some protozoans. And many of those eukaryotic algae, when they arose with chloroplasts. All right, so chloroplasts for just a second. Let's take a look at this next slide. So here it is, about 2.2 billion years ago, we get this big, look at this, this is what's called the oxygen revolution. And that's all because of concentration of these photosynthetic organisms. So now when the oxygen revolution came around, then a life that was a little more complex, the eukaryotic life, could then take advantage of that excess oxygen. Right? So the oxygen that these photosyntheses were producing was drifting off into the atmosphere. It was killing a lot of anaerobes that couldn't deal with it, but there were some critters that could make use of that oxygen, could make very good use of that oxygen. So this is when aerobic respiration would have started to come on the scene. So again, this is back to that getting 2 ATPs versus getting, nearly 40. So at that particular time, you're going to get this, what it says here, the rise of eukaryotic life. You see, what we find so many times when we study this is that one organism prepares the path for the next organism. Now, they're not doing this with a thought of, helping the plants helping out the animals. It's not done that way, of course, but it's sort of like a random thing, like an evolution. The rising oxygen levels allowed more complex critters to arise, the eukaryotic organisms. What happened to those anaerobic prokaryotes, the ones that didn't die off as these oxygen levels begin to rise? Well, many of them went extinct. A lot of them went extinct, but some of them survived by actually hiding, if you will, or holding up inside of another cell, something we call endosymbiosis. Now, symbiotic or symbiosis is organisms, living together, right? And endo, of course, means organisms living together, one inside of the other. So a symbiotic relationship, we've mentioned a couple of them, could be mutualism, predator-prey is a symbiotic relationship. Here we're in a symbiotic relationship where the partners, one partner is inside the partner, is inside the other partner. Let's take a look at how that might have happened. About 1.2 to 1.8 billion years ago, we start to see this rise of multicellular eukaryotes, possibly this rise of multicellular eukaryotes. And this, of course, as I mentioned, allows for a lot more greater complexity. And you can now make organelles too. And those organelles could be the division of labor, right? And that division of labor would have made that thing more efficient, and that would have been driven by evolution. These eukaryotic cells also had a cytoskeleton. Remember the microtubules, microfilaments, the intermediate filaments, all those things you learned about in Bio 1 that do all those different things. They give structure to the cell. They keep the cell from being torn apart. Organelles will move down parts of the cytoskeleton as they're making their way around. But another thing that the cytoskeleton allowed cells to do, it allows them to engage in endocytosis. I know that word's familiar to you, and if it's not, there's a picture right below to show you what I'm talking about. What does it mean when we say a cell engages in endocytosis? What is endocytosis? And if you're thinking, oh, it can be big or small, you're correct. But what does it mean to endocytosis something? Okay, Mark, yeah, it can be used to build up things, but what is the cell doing when it's engaging in endocytosis? It's building up something, it's accumulating something, but what physically does the cell do in order to engage in that? Anybody else? Look at the picture. What's going on in the picture from A to D? You saw a picture like that very close in your bio book. I know you did. Remember, endocytosis can be, yeah, not making a new cell. We're going into the inside of the cell, creating a vesicle. We're creating a vesicle by engulfing, right? So in my little picture here, if this cell wanted to engulf these particles, it could do that by using its cytoskeleton to move its membrane around to pinch off and form a vesicle. And Mark said, what, make a build up? Yeah, you're building up a concentration of those materials. But remember, endocytosis could be large particles, which is phagocytosis, and then smaller particles, which is pinocytosis, right? So we got this engulfing things. Now, prokaryotic cells are not known to be big engulfers. They're not engulfers, but eukaryotic cells are engulfers. That's why you learned about endocytosis in Bio 1. It's one of the big things that eukaryotic cells do. So let's go back to our question about some of those anaerobic prokaryotes that were trying to get away from the oxygen. One of the ways they could separate themselves from the concentration of the oxygen was to be inside of something else. And we got cells out there, these eukaryotic cells, that like to engulf things. So what the endosymbiotic theory tells us, or provides us, with a theory or an idea of how certain organelles, certain really special organelles that we find in eukaryotic cells today came about. So if we start in the upper left, we have this ancestral prokaryote, and through time, this ancestral prokaryote developed the ability, as it became a eukaryote, to engulf things, right? So it begins to engulf particles, food, and all that sort of stuff. In the picture you see here, it's engulfing an aerobic bacterium, maybe similar to the one in the lower left, that fossil cyanobacterium. And in the other one, we're engulfing a photosynthetic bacterium, kind of like what you might see in that mat of green algae in the upper right. That's actually a prokaryote that's making that. All right, so we're engulfing the bacterium. We engulf this photosynthetic bacteria. Now, if you engulf a photosynthetic bacteria and you don't eat it, you don't destroy it, you become symbiotic with it, what advantage did the eukaryotic cell that engulfed that photosynthetic bacteria, what advantage did it now acquire because of that process? What advantage was conferred to the eukaryotic cell when it engulfed a photosynthetic bacteria? What could it do now? Produce food. Yeah, perfect. How about when it engulfed the bacterium? And if I tell you that the engulfed bacterium eventually became the mitochondria, what advantage did it get from that? Gotta go back and remember your respiration chapter. Mitochondrium is the powerhouse of the cell, right? Now, you don't create energy, Mark. I'm sure that's probably, you didn't mean to say create. You only convert. But what about the conversion of energy, the energy in food, to the energy in ATP. What about that is important in context of the mitochondria? If you don't have a mitochondria, which step of respiration are you limited to? If you don't have mitochondria, what part of respiration what's the only part of respiration you can do? That's it. That's right. You can only do, because that happens out in the cytoplasm. You know, everybody does glycolysis. And how many ATPs do you get from glycolysis? 2. And how many do you get if you have glycolysis and the rest of the process, which needs a mitochondria to do that? How many do you get out of that? Yeah. So having that engulfed aerobic bacterium, it's part of the symbiotic relationship, as well as the photosynthetic one, is it might get protection from these high oxygen levels, protection from other organisms out there that would want to eat and destroy them, and in turn, the photosynthetic one allows the cell to photosynthesize. That's a heck of an advantage. And then the aerobic bacterium allowed that cell to crank out 20 times more energy from the same glucose. Now I ask you once again, would those have been things typically favored by evolution, those two engulfing of the photosynthetic and the aerobic? Do you think evolution would have favored that? And it would have favored it. Why? What did we just do to the cell by giving it a mitochondria, or in some cells that have both? Yeah, there you go. Efficiency. We've made them more efficient, and that's what drives natural selection. Good job, guys. All right, so once we have this complexity of cells being formed, these eukaryotic cells, we've actually made our way now through time to the Cambrian explosion, about 500 million years ago. And this was the thing that Darwin had such a problem with. If you look at the chart down below, before the Cambrian explosion, so we're looking, you know, somewhere about right in there, there were a few different groups of animals. Animals were around prior to the Cambrian explosion. See, the Cambrian explosion is this line right here on the left. So the animals were around before that, and this is what bothered Darwin. After the Cambrian explosion, we had this rapid proliferation of life forms, especially all life forms, but most notably among the animals. The sponges were around, the cnidarians were already around. Those are the jellyfish. Some of the other critters were around, but after the Cambrian explosion, we just have this rapid, that's what he saw in the fossil record, remember? He's digging through the fossil record and he sees, a few creatures, and then all of a sudden, in a relatively short period of time. Now, for Darwin's idea, or for Darwin's perspective, it seemed like a short period of time, but we've actually done more investigative work and found out that this Cambrian explosion had a pretty long fuse for an explosion. The Cambrian explosion exploded, if you will, over between 25 to 100 million years. So it wasn't quite as fast as Darwin might have thought, but that still is pretty fast. Still pretty fast to go from basically a few handfuls of some simple critters to basically all of the modern critters, not the species, but the groups, okay, like the chordates and the echinoderms and the annelids, and we'll talk about what all those groups are as the semester goes on. So I'm not saying every species arose back then, but all these major groups arose. The reason why we think, or one of the major reasons behind this Cambrian explosion would have been an arms race. At about the time the Cambrian explosion occurred, eukaryotic organisms were growing larger, and they were growing the ability to feed on each other. So these eukaryotic critters were developing claws and abilities to capture and consume prey. So whenever you have something like that, whenever you have a strong predation pressure, you exert a very heavy selective pressure on everybody around you. The ones that don't want to get eaten, they're going to have to figure that out pretty quick. You got to either move faster, camouflage, fight back, protect yourself. And as soon as they find a way to protect themselves from the predator, then the ownership is back on the predator to change. Because if the prey organisms get really good at not being consumed by the predator, then the predator go extinct. So you see, it's an arms race, right? Back and forth. Prey develops a way to protect itself from the predator. Predator says, well, heck with that. I develop a way to counteract what the prey is doing. And then the prey says, okay, well, now I got to figure out some way to protect myself. Now, along the way, one of them could go extinct, right? That's typically what's going to happen. But if you do survive and you do continue on with this arms race, then that back and forth is going to occur. And so we think that one of the things that contributed to this explosion during the Cambrian period was this arms race, this ability of organisms to begin consuming one another, to begin attacking, killing, eating one another. Now, soon after that, the land at this time is barren. And soon after that, organisms began to move out on the land. But it wasn't animals. Again, animals didn't have anything to eat. It was the plants that first moved out on the land. But even though plants need dirt and soil to live in, the dirt in the soil back then wasn't very nutrient rich. The kinds of nutrients that plants need today that they find in the soil weren't all that abundant in the early soils on Earth. So when the plants moved out on the land, if they'd done it by themselves, a few species might have made it. But for the most part, most plants wouldn't have been able to survive. Plenty of light, yes, no problem. Water might not have been an issue, but the nutrients that need to be absorbed from the soil that allow that plant to survive, there wasn't that much nutrients in the soil back then. So we believe that plants actually moved out onto land together along with a fungus that we call a mycorrhizae. There's mycorrhizae. Mycorrhizae is a fungus. Now, we'll learn a little more about mycorrhizae when we get into the fungal chapter, but let me show you what a mycorrhizae fungus does. Next, I'll come back to the previous slide. This is a cross-section through the root of a plant. So, cross-section, right? We've cut through the root of the plant, and you can see on the inside the cells of the plant. These are the cells of the plant. And then on the outside, what we have is that mycorrhizae. This is the fungus. So mycorrhizae, if this is the root, makes a layer around the outside of the root. Now, depending on what kind of mycorrhizae it is, it either can penetrate down into the cells with its little feelers. You can kind of see that in the larger picture, or it can actually penetrate down in between the cells. Regardless of how that happens, it actually does not damage the plant, but what it does is it allows the plant to extract nutrients from the environment more efficiently. We're going to learn when we get into the fungus chapter, fungi get their food very much different than the rest of us. Plants make it, we consume it, bring it into our bodies and digest it. Fungi digest their food on the outside. They actually secrete enzymes, digestive enzymes, into the environment, dissolve the nutrients, and then soak those up. All right, now, think about that in terms of a plant. A plant can't get at those little nutrients that maybe the fungus can. So the fungus can start dissolving those nutrients. Some of it uses for itself. Obviously, that's why it's doing it. But it's like plants make more food than they need. That's why they grow. So a lot of those dissolved nutrients that the fungus has dissolved is more than the fungus needs. Now the plant can have them. All right. So the mycorrhizae actually produced a much more conducive environment to plant growth because it allowed access to those nutrients that might have been otherwise tied up in the chemical compounds of the soil and the dirt that it was growing in. Now, we believe this is so important because today, modern plants, about 90% of all species of modern plants have mycorrhizae. They have these symbiotic fungi that live with them. So we believe that was probably a pretty important thing. Soon afterwards, the animals followed. First was the arthropods. About 450 million years ago, the arthropods began to move out on the land. They're really good at, arthropods are good at surviving dry conditions. And we'll talk about that when we get to them. But they would have been some of the first ones. And then, of course, when the arthropods moved on the land, they were great for the plants because they could move the spores and all the stuff that the plants were using to reproduce, and then later on the seeds and all that kind of stuff. And then the arthropods provided something for something else to eat, right? Plants oftentimes, moving on the land, would have developed these waterproof coatings on their leaves to prevent them from drying out. Even today, there are some plants that way. Red mangroves, if you touch the leaves of red mangroves, they have a thick waxy layer on them. It prevents them from drying out because they live in partially salty water. But in essence, you know, with the arthropods and everything else, plants created new opportunities for everything else. You know, once the plants came out on the scene, then lots of other critters could evolve. You know, the animals could come out onto the land and they had something to eat, and then evolution, then an arms race began on land and the plants evolved. So it was really at about this time, this 500 million years ago, when this rapid proliferation of different types of life began to occur. Not just in the animals, but in the plants. See, the plants came just a little bit after the Cambrian explosion, the land plants. Plants wrote here, the land plants. It hasn't been all hunky-dory since then, though. The Earth has actually gone through a series of mass extinctions. 5 or 6, I've heard, recognized mass extinctions. We're going to talk about 2 of them. We'll talk about the Permian and the Cretaceous. But if you look at this chart, the red line is the extinction rate. The blue line is the number of families. So we've had, we got 1, 2, 3, 4, 5. They got arrows underneath them. So we had an extinction rate way back in the Paleozoic. This one in the middle, this big one in the middle, that's the Permian extinction we're going to talk about in a second. And then over here, the last one, 65.5 million years, that's the dinosaur extinction. So the Permian and the dinosaur extinction, the Cretaceous extinction are 2 of the biggest ones and 2 of the most recent biggest ones. But notice, in spite of the extinctions, the number of families keeps growing, right? Keeps going up. Extinctions are bad for the critters that are going extinct, no doubt, but they're not necessarily bad for the other organisms, because sometimes extinctions open up opportunities for other critters, right? Kind of like when your parents go out of town and leave you to the house to yourself. It opens up opportunities that wouldn't be there if your parents were there. So these extinctions could maybe, for example, wiped out a group of predators that was exerting heavy predation pressure on this prey. Now the predators aren't there, so the prey obviously have the place to themselves. Okay, so let's look at these extinctions. Let's go to the Permian one first. So here we are, about 250 million years ago, we get the Permian extinction. Look at how incredible this was. 90% of all the marine species went extinct. A third of all the insects went extinct. And this happened pretty quick. This is like that punctuated equilibrium thing I was talking about. It only took about 5 million years for it to happen. Extinctions and major geological events oftentimes are what we use for the boundaries between these eras. See, the Permian, there's the Permian at 250. The Permian extinction was the boundary between the Paleozoic and the Mesozoic. And it's not just an arbitrary designation there. At the end of the Paleozoic period, the opportunities that were opened up allowed the dinosaurs to rise to prominence in the Mesozoic. Remember, the Mesozoic is the dinosaur era. But before we get to them, let's talk for just a second about how this extinction came about. Well, one of the things that may have happened during this time, or one of the things we believe happened, was the formation of the giant supercontinent of Pangaea. When land began forming on Earth, it didn't form in the arrangement it is now. It actually formed as one giant single continent, Pangaea. Now, I'll come back to the Siberian volcanic eruptions in just a second, but let's take a look at Pangaea. Everybody, I'm sure, is aware that the Earth is a series of these crustal plates that float on a semi-liquidy mantle underneath. And these plates are moving around Earth and bumping into one another. This is Alfred Wegener's theory of continental drift is what this is, okay? And these plates bouncing into one another led Wegener to come up with this idea of continental drift. Now, he didn't realize it was the plates that were doing it, but his observations came from, because if you see it like a puzzle, like South America and Africa, if you look at them, they look like they could almost fit together. And they do. Right? They do, especially when you look at them at the real, the continent. The real edge of the continent is underwater. So if we start including that part, then we see that the fit is actually a lot better. So with these plates moving, and the arrows are showing which way they're moving, like the Atlantic is getting wider and the Pacific is getting narrower because the North American plate and so on and so forth. But with these plates moving around, these continents moving around, we can go back in time And we can say, all right, there was a time in the past, about 250 million years ago, that we believe Pangea arose. Now, when I mean arose, it didn't come up out of the water. There were all these little continents, little land masses, but they finally arose enough to make this one giant Pangean supercontinent. Now, when that happened, that would have caused massive destructions among a lot of marine organisms. The reason why that is, here's another picture of Pangea. The reason why that is a lot of marine organisms live near the coast. They live near continental coasts. Even today, there's not a lot of life out in the middle of the ocean. A lot of the life occurs around the coastal part of the ocean. And the reason why that is, that's where the food is. If you think all the way back down to the producers, the plants, you know, they can't grow in a mile deep water. They have to be near the coastline. So that's where the food is. And that's where the critters that eat the food and the critters that eat them are going to be found, right? That all makes perfect sense. When Pangea was formed, the amount of coastline was decreased. The amount of coastline, well, imagine all these little tiny spots of continents, and then they finally formed together in this one supercontinent. You actually are getting rid of a lot of the coastlines, a lot of these inshore waters. The ocean got deeper. The inshore waters were reduced in quantity. And so what we have is a situation where we have less habitat for a lot of marine species. That's why a lot of them would have died off. Another thing that happens, and this would have been actually something that led to the next group of species, was you'll notice that as this continent formed, areas inside the continent were ranged further away from the coast, so they started getting drier. Now, think about it. The first critters that moved out onto land, amphibians and those kind of critters, would have needed moist conditions to survive. Amphibians definitely, because they need eggs, and their eggs have to be laid in water. But if there was an organism, a critter, a species, or a group of species that could survive in these dry climates, in other words, didn't have to go back to water, maybe came up with a different kind of egg, for example, then they could have started dominating. Now, what critter do you know lays an egg that doesn't need water and is good at living in relatively dry climates? It usually has a pretty protective skin to keep them from drying out. What group of critters is good at that? So amphibians have to lay their eggs in water. We don't lay our eggs in water, us mammals. Birds don't, but there's, yeah. What's a critter out there that doesn't need water to lay eggs? It can lay its eggs out of water, doesn't need water, and it generally does pretty well in drier climates. There you go. Reptiles, right? And of course, who's the king of all the reptiles from history? Sure, the dinosaurs. So the formation of Pangea would have destroyed a lot of the marine species, but provided a habitat for the reptiles to come along. Now, I'm going to show you the dinosaurs in just a moment, but let's go back to this idea of this massive volcanic eruptions. Because these massive volcanic eruptions, you know, the Earth was volcanic, then it cooled off, but then there could have been these other periods of heavy volcanic activity. I mean, the Earth hasn't reached that completely cooled off point that we have now, where we have very few volcanic activities. And this is a map. I'm sorry, it's in Russian. It's the only one I could find. This is a map showing the extent of, during the Permian time, what they believe was a volcanic lake. So the area in the blue that's outlined, not volcanoes. I didn't mean to say volcanic, like I meant to say lava lake. So a lake of lava, just this massive, it takes up half of Siberia, for goodness sakes, this massive lake of lava, all this molten rock, and all those gases, those hydrogen sulfide, and those toxic and bad gases were being dumped off into the atmosphere. So the Permian extinction might have been a little bit of both, right? Might have been, or probably was a little bit of both. The collapsing of the coastal area where a lot of species are found, and then all this volcanic activity was going on. I mean, can you imagine walking across Russia back then, you know, cruising across, and you come up against this, I guess we could do the miles, that's 1,000 kilometers, so we're talking 4,000 or 5,000 kilometers across. The blue line by Scandinavia, you mean the one I just drew? Is that what you mean? that what you're talking about, Thomas? You see that right there? The coast, I think they're just trying to show the coastline, I guess. The squiggly line is the extent of that volcanic lake. And if I could read Russian, I could figure it out. Is that what you're talking about? The blue there? Yeah, that was, I think they were just trying to enhance the coastline there. Yeah. Okay, so after the Permian extinction, here they come, gangbusters, right? And they rose to prominence during the middle. Now, the big dinosaurs didn't come around until about the middle of the Jurassic, basically. Initially, the dinosaurs were small. Yeah, I think this is cool. I like that picture because it's like everybody coming at you. You know, it's like if they all evolved at once, and here they come. And of course, the dinosaurs kicked everybody's butt, right? Because the dinosaurs were mean and bad, and you know, they were reptiles, and they could live in these really sparse conditions, in these dry conditions, and they were doing really well until the Cretaceous extinction. 65 million years ago, about half of marine species went extinct. We lost most of the terrestrial plants and animals, and all the dinosaurs. And this extinction occurred, we believe, because of an asteroid strike. You see the picture in the upper right. And the reason why we believe it was an asteroid strike, this is known as the Alvarez theory, is if you look where I've put the red arrow, if we dig down through the rock layers, when we get down to the rocks that were laid down about 65 million years ago, we find this really thin, you can see it in the picture, this black line. We find this really thin layer of an element called iridium. Layer occurs all over the earth, iridium. So this is a thin layer of an element, iridium. Now, iridium occurs on earth, but it's very rare to find it on earth. So where would all this iridium had come from that enough to basically circle the earth? Well, iridium is rare on earth, but common on asteroids. So it could have been something, you know, here comes the asteroid, strikes the earth, blows up, all that iridium, you know, and including all the other stuff, gets, you know, circles around the earth and eventually falls back down to the earth and forms that layer of iridium. The smoking gun of all this for many years was, where's the crater? An asteroid this big, by the way, the asteroid is about the size of Manhattan. An asteroid that big should have left a crater. And we actually did. The Chicxulub crater near the Yucatan Peninsula. It actually extends, it's partially on land and partially in the water. But we've actually been able to find a crater that we think was the crater caused by that asteroid. Look how close Florida is to that. That would not have been a nice day. And this is similar to what I showed you when we were done the first time around. This is the progression of life, you know, after we've gone through all of this. You know, we start down here with the formation of the earth. So this is just a summary. What I like about this chart, I would like you to focus on when you're looking at it, is compare what's happening in the animal world with what's happening on the world geographically. So this map on the right. All right. Where are we? Now, of course, the reason why we get such rapid speciation too is for reasons: punctuated equilibrium, adaptive radiations, you know, the ability, once the dinosaurs were extinct, then that opened up new opportunities for other critters. And like, for example, amongst the mammals, we have this worldwide radiation of mammals. You know, few mammal species really look at what happened after 65 million years ago, though. Here's about 65 million years. Look how big. Now, the monotremes we'll talk about, they're weird. They always kind of, they never really diversified. But the marsupials and the rest of the mammals certainly did. So it could have been worldwide adaptive radiations, local adaptive radiations, or regional. This is the adaptive radiation of a species of plant. A close North American relative is the tarweed. And these plants that you see have all evolved out of that close North American relative. And they've moved throughout the islands. You can see about 400,000 years ago, Hawaii, we start to get this species, Dubadia linearis. And all of these are related to one another, and they're all different for the same reason that those finches are different on the different islands, the different Galapagos Islands. So after the dinosaurs left, it was a party. Everybody was adaptively radiating. Everybody was moving out, moving around the world. Let's talk about developmental genes. Developmental genes are genes that would affect things like the rate and the timing of things that grow, or the spatial pattern of things that grow on an organism. So these are the genes that are active at the very beginning of the organism's life. So if you make slight little changes to the gene early on, you can have some very dramatic changes that occur after that organism grows up. And the two things we're going to look at heterochrony, which involves the rate and the timing, and then we'll look at homeotic genes, or spatial patterns. So let's look at heterochrony first. Heterochrony might explain this, for example. If we look at a chimpanzee fetus skull, we look at a human fetus skull, they look very similar. But of course, when they become adults, they're very different. What we think would have happened here is different developmental genes would have controlled the rate and the timing at which different parts of the skull would grow. So whereas the adult skull, the jaw, the rate at which the jaw grew would have been slower than, say, in the chimpanzee fetus skull. So the jaw of the chimpanzee is much more larger and pronounced, and the skull would have sloped back more than it did in the adult human. So just looking at these two species, you can see that if you change developmental genes in organisms, especially at the beginning, that what you would get is dramatic changes later on. So it's kind of like, the penguins and the March of the Penguins going across the Antarctic continent again. It's a long way to walk 70 miles, but you can do it 6 inches at a time. So if you take these little tiny changes at the beginning in rate or in timing, you can have profound differences once that is extrapolated later on to the adult. Now, what heterochrony can do as well is heterochrony can actually create organisms that look like a juvenile, but actually can reproduce. Neoteny is the process by which you change the timing of the reproductive growth compared to somatic growth. Somatic growth is growth in the non-reproductive parts. Eventually, through time, if you change the timing of those two things, what we're saying is we're changing the timing in which an organism becomes sexually mature. So if you eventually do that, what you might end up getting is ancestral juvenile characteristics in an adult, in a descendant adult. This is called paedomorphosis. And eventually, if that paedomorphic organism that has those ancestral juvenile characteristics is able to reproduce, it now engages in paedogenesis. Now, let me show you an example of where that happened. This is the axolotl. This is a salamander. It's got gills. This is the adult. Young salamanders also have gills. Young salamanders, juvenile salamanders look like this, but this is the adult of this particular organism. We think that the axolotl grew into an adult form and the ability to reproduce while retaining these juvenile characteristics because of changes in the rate or the timing of the genes, and because of heterochrony. Same thing with the slimy salamander, which is a Florida species. We think they came about and look the way they do. They look like juveniles, but they do have the ability to reproduce. So you get a whole different form from that. You get juvenile-looking organisms that aren't really juvenile. They're only juvenile in morphological appearance. They're not juvenile in terms of the ability to reproduce. That would cause little changes in the genes to do that. Now, as far as the pattern, the spatial patterns of different parts of the body, there's a class of genes out there called the Hox genes. Hox genes are a type of homeotic genes. Homeotic genes control position in an embryo, where the legs grow, where the appendages grow. So, for example, if we look at crustaceans, the two crustaceans down below, look at crustaceans, the two arthropods down below, the crustaceans on the right, Artemia is a crustacean, Drosophila is an insect, but they're both arthropods. And they have these Hox genes. And in this work that they've done, they found out that a mutation in this one Hox gene, the UBX gene that you see there, UBX expression does not suppress leg formation in crustaceans. See, crustaceans first started out with making lots of legs. But as time went on, there was too many legs, and more advanced insects have less when it comes to appendages and legs. So what we think happened about 400 million years ago was a mutation to one of these Hox genes that actually began to suppress leg formation, and that's how insects came about. So these combinations of heterochrony, changing the timing of genes, and this modification of a developmental gene that gives rise to appendages and is being mutated could have taken us from Artemia, which is still around but has lots of legs, to Drosophila. Now later on when we get into arthropods, we're going to see that appendages, it's all about appendages in arthropods, different types of appendages. And these modifications or these mutations to these Hox genes, to these positional genes, would have led to all of these modifications of arthropod appendages. What make arthropods, one of the things that makes them so diverse. You know, there are pinchers, and there are they even have appendages that they can sense, they can smell and sense taste with. So more of that when we get into the arthropods, but we think that that's probably come about because of these homeotic genes. Now, I'm not going to go through horse evolution. That's not my point of this slide. What I want you to show you, that evolution does not have a goal, right? There's no goal. We're not, you know, the goal of evolution way back 55 million years ago when we were dealing with the Hyracotherium, the relatives of horses, wasn't to make the modern horse. You notice there's all kinds of horses through time. Each one of those things is the name of a horse, and not all of them survived all the way. Like Myohippus, somewhere around 37 million years ago, died off somewhere around, say, 18 million years ago. But notice again, it's going out like a branch. It's not a linear chain. That's what we mean by evolution does not have a goal. And even if it had a goal, the Earth changes. So you can't have a goal if the goal keeps getting moved. Now, I know I'm at time, folks, but I ask if you would just give me another couple of minutes to finish up this last part, because I got to finish this chapter so you have the information for the exam next week. What we're going to talk about for just a few moments here is how do very unique evolutionary novelties come about, like eyes and feathers and legs. Stephen Jay Gould, who was a evolutionary biologist at Harvard, used to write an article, and one of the things he mentioned in the article was talking about this, and he kind of asked this rhetorical question: what good is 5% of an eye? The eye that we have today is a very complex structure, and it couldn't have evolved all at once, right? But Stephen Jay Gould says, what good would only part of an eye do? 5%, 10%, he's just using a number. It wouldn't function. Now, what we're talking about here, these things like eyes and feathers and legs, are called exaptations. These are structures that evolved for one purpose, but they ended up becoming fortuitous, evolutionarily speaking, in another context. All right, so eyes didn't start out as eyes the way they are today. Legs didn't start out as legs the way they are today, and nor did feathers. So let's take a look at eyes, and we'll mention legs and feathers actually when we get to organisms later on in the semester. So let's follow through from A through E here about some primitive eyes. But let's not call them eyes. Let's say they're light sensing devices. So up here in A, there would have been an organism. There are even organisms around today. A limpet is still around today that wouldn't have had an eye, but it would have had photoreceptive cells. Now, would that have been an advantage to be able to sense light? If you were a limpet, yeah, Thomas, if you were a limpet and things fed upon you and you were living at the bottom of, say, a body of water, you might be able to sense when a shadow passes overhead. Now, you don't know what the shadow is, but because that shadow passes overhead, you might move off into some place where it's a little, where you're a little bit more hidden, right? A little camouflage. That would have been an advantage. That would have been preserved by Mother Nature. Now, what you see happening in B through E, and again, this isn't an A&P class. I'm not going to talk about the specifics of the eye, but the eye is becoming more advanced. For example, in B, we make an eye cup. So now light can be focused from different angles. The object doesn't have to be right in front of you like in the pigmented cells. And then if you actually cause that cup to narrow in and make a pinhole eye with a fluid-filled cavity, what you start to do is you start to increase the resolution of that image. Now you can see it as more than just a shadow. Now you can make out the shadow and then add a lens and then add a complex lens that can be focused. So you see eyes down here at the right, Lolligos of squid. Complex eyes didn't evolve all at once. They evolved through time, and each time they conveyed a selective advantage to the organism, their ability to sense light, right? Their ability to see. So early chordates would have had an eye spot. Later on, animals may have had light sensing cells that actually bulged out so they could see up and down and around them. And then the cup would allow them to focus light. And the focusing of the light means light can come from different sources, and it gets focused on a retina. And then when you add a lens, you finally can get yourself to a clear image. Okay? So eyes were fortuitous at the beginning because they weren't eyes. They were just light sensing organs. And they eventually evolved into eyes. And again, we'll talk about feathers and legs later on when we talk about those critters. Sorry, folks, I ran over a few minutes, but that's it. I'm done. Let me turn the recording off.